Now we have almost an hour which we can use as we would like, in the sense that we are here not to read but to discuss. And the more the more we get into our common interests and exchange ideas, the more valuable it will be. Yeah. So we will have 4 members of the panel. 4 members of the panel. Then this is Turgut, who is a part of the consortium. And then we have some kind and friendly and critical external partners or external evaluators. And it is again Martin Wolf from Zurich, who is also a technologist, I would say, and Heidrun Wabnitz from Berlin, also on the technical side, and then there's Adelina Pellicer, who is a neonatologist from Madrid. But then we have all you who have chosen to spend a morning here. And before we go any further, I would like to have a brief show of hands. How many of you are on the medical side with a medical background and medical interests, like myself and like Adelina? So how many have a medical? Monica is there, Agnes, I recognize you, and Björn. So medical people, we are in a minority. So, so we will have to be careful not to sort of make ourselves thrown out. Yeah. How many have a technical background? Technical physics, much stronger representation. How many have a kind of a business management project managing? Yeah. Okay, so the clinicians, at least we are more. I mean, that's, yeah. Yeah. Then I wanted to say that I think the problem is that on one side we have physics. We have the speed of light, we have an energy of a photon, we have the basic physical laws, and I don't think we are going to change that. Then we have technology. And technology, we live in a society where technology improves at an incredible speed. And then we have the body. The human body with physics, its physiology, blood that is flowing and blood vessels that are moving. And then we have illness, we have problems, we have what we call pathophysiology, when this body as a machine, this machine is broken. Again, I think this is a part of nature. We are not going to change that too much. Then we have medicine. Medicine in terms of making diagnoses, classifying things, and also doctors like to do things, do treatment. So you can say medical science, medical knowledge. And then in the extreme other end, we have healthcare practice. I mean, what does doctors and nurses and hospitals and hospital administrators, what do they do in real life? So we have this line from physics to the social machinery of caring for the ill. Yeah. So I'll just like our panelists now to say, where is the problem of this machine on that line? Where is the problem of this machine on that line? Because when we speak of changing, I mean, working in order to improve things, we have limitations and then we have challenges. I mean, limitations, that is something we can't change. Challenges, that's something we can address. Then we have opportunities, and probably we get most done by sort of addressing the opportunities. So I'd like our panelists to have the first bit, and then I'd like all of you to think of this and then be ready to play in when, yeah. So, Adelina, you are ready?
Thank you. Thank you for inviting me to come here. It's a very interesting opportunity. As I see, we've been in the last probably 20 years trying to implement near-infrared spectroscopy in a clinical routine, in our clinical routine. And I would say that currently is a trend monitoring. So that to have, in the scale you have defined, maybe the best the best position I would leave for this kind of monitoring would be in the physiology or pathophysiology studies. I mean, a very clear, defined scenario where you really control when the intervention starts and to see what's happening. And I say that because the most popular and the highest recommendation to use in a clinical setting for this kind of science of physics is among anesthesiologists in the operating theater, where it has been really demonstrated that makes a difference. Even the evidence is not high enough, but the recommendation is very strong. So then we want to translate this in our routine, which is the society, you say. Can this kind of instruments help us really to get better results in our patients, in day-to-day care interventions, not in a very precise intervention? This is still to be defined, so we really don't know. And then regarding this kind of instruments, I see a problem. The problem is that currently is too big and is too hard. And this is in contradiction with what we really try to do in our routine, which is allowing the link between parents and infants, being putting less things, complex things on their heads or whatever. So I would really, I think that on the one hand we need to exploit really this kind of technology. We need to be sure that we need that to get better results for sure. I think are extremely promising. We need to make it more simple to be used in a clinical setting. And now I think their role is mainly on interventions that are defining pathophysiology, very concrete interventions. That's my point.
No, not now.
Okay. But despite your question, I would like to start with something positive. I think the original idea was that if we just measure oxygen saturation like we do that today, then we can see changes, but we cannot really determine what is the origin of these changes. Is that a different blood flow? Is it a different oxygen consumption? And I think that was the original idea of this project, was to sort of provide the parameters so that we can really determine the oxygenation state of the brain of these newborn infants. So we now, if the saturation changes, we know it's oxygen consumption, we know maybe it's blood flow, and I think this should be clinically useful, I would guess. I would expect that this in the future will be clinically useful, and I think from that point of view this project is quite a success. And then there are some problems that I hear about the heart sensor, and I understand, because we did a lot of measurements in the clinic as well, that this is a very important issue, but I think this is something that can be solved by, this is basically an engineering problem. I think that you can solve. Miniaturization is also a question of the money in the end. So yeah, I think I'm pretty positive about this project.
Should I say something? Okay, so I think with my background I should say something about accuracy. So this is another issue where I think the project has made some progress. You have shown that. It's not like orders of magnitude improvement, but a factor of 2 is something already. I remember your presentation, I don't know, many years ago at a Royal Society meeting where you said the usefulness of this method depends on its accuracy. And that's why I think we have to make progress here, and also we have to better understand what accuracy really means. And here you were mentioning the calibration issue. For my understanding, the TRS method allows us to get such values without the absolute need of a calibration. But what we definitely need is to validate, to check whether the value we get is consistent with other measurements. So this is definitely a step which has to be done. And also, so I'm pretty much involved, like many of you in the meantime, in this standardization efforts in the cerebral oximetry. And I think what you have shown here and what the consortium has achieved is another contribution to that discussion, to finally to improve on that and to avoid, also to check which accuracy is achievable, but also to avoid a false accuracy statement. Yeah, thank you.
So I am a physicist. I'm one of the technology developers, and I think everyone expects me to wear that hat when I speak here. But I will try to be a bit controversial, and I will wear a different hat. A hat of someone who's missing from this table, I think, at least this group who's speaking, which is a patient's hat. So I had the unfortunate situation of having a child who spent about a month in the NICU about 2 years ago. And I always liked the standard to which we are held. So we should have a very soft probe, it should be very easy to use. And for a month I watched how you put pulse oximeters on a baby, what happens when an ultrasound comes, what happens when an X-ray machine comes, and I have learned that where the standard is and where the practice is, there's a big gap in it. And when I view BabyLux from that perspective, I feel that we are not that far from being usable. It's not the ideal, but when I see how long it takes to put a pulse oximeter on the feet of a newborn baby, premature baby in the NICU, and how it's an important device, I think we are not so far from it. So I would like to view that as a positive from the patient's perspective who was looking at it in a critical manner. So what we have to weigh here from that perspective to me is that at what point can we be convincing enough in our data that if we took this data from a much larger group of people, we might be able to give you information that you can make decisions that can improve the lives. And then, of course, later on we have to look at it, how fast this could be done and how well it could be done, and where we are in comparison to all these other devices. So I think we should bring this aspect to the discussion as well, to really go into these rooms and look what's being done to these babies and how it is being done, and where the standards are, and at what point can you sneak into that practice. I don't know if that was the answer you expected.
Agnese.
I am a medical doctor, young medical doctor, so from my experience it's a really good idea, and it can answer to a really important question that we have. We have no other parameters for blood flow, for example, or not feasible. But in our experience, I confirm that the technical problems were many, and so from one part I think that we cannot stop to find a solution, because our question from medical doctor is a very big important question, but the technical problems are many. For example, another one that I saw is that we were not able to carry the continuous monitoring. And I think that for the brain monitoring, we need a continuous one. I mean the 24 hours monitoring. We did not do that because it was not feasible. And this is one problem for me.
Our boss.
Okay, thank you. No, I just want to remind all the people here in this room that we were not allowed to do any research in this project. So the device you see here is a device that is made of components, technology, ideas that were ready 3 years and a half, almost 4 years when the project, or 5 years when the project was devised. So. But the important thing is that having this device right now, maybe 1, 2, now we have 2, maybe 4, 5 devices in different hospitals is important because we can get clinical results, clinical research results, let's say. But then we have already started new projects for miniaturizing the technology. So if I look ahead of, say, 5 years, 10 years, that device can be as small as a box. And also software. Not sure it will be cheaper. So this will be another point.
Yes. I can continue with Turgut's comment. How can this be used to improve the outcome? We really don't know. I think that one very important achievement of your device is that precision on saturation, to measure tissue saturation, is really very good. Because to me, I've been using infrared spectroscopy for many, many years, and I still think on this technology as a trend monitoring, even that it provides absolute values, but as you see that the precision is not as good, and differences among the different brand devices you use are sometimes not small. So my impression is that it's useful to take care of what our interventions induce in a patient in a given moment, but I don't trust so much on the current absolute value. So your achievement is really right in terms of precision. So the question is, would this property of your device allow us to take reference values, use threshold values? That's very good, because the precision is very good. This is an achievement that I would remark here. I'm not as convinced with the other parameter, with blood flow, because this is semi-quantitative. It's not blood flow. It's a semi-quantitative assessment. It's kind of what we get with Doppler. I understand that this is a continuous measurement. It's not an intermittent measurement. But still, we need to know more about that in a clinical setting. Again, I think the precision is good enough, as Gorm said, because I had my experience on indocyanine green to look at the blood flow index, and I mean, it was 25 to 50% variability, so this is very good, even it is around 22, 25%. So again, promising, but Gorm, I'm not sure what it really means in terms of physiological, the interpretation of that, because it's not really, as you said, it's not blood flow, it's an index. So I don't know if at the end is also a measure that we will be able to use again as a trend. I don't know.
So this is always an interesting question, because I would like to turn the table and ask you a question about this. I've spent the last 20 years from my PhD time working with people who make blood flow systems, so inventors of ASL and everything. And as far as I know, in every technique, there's an algorithm where something is hidden, you get a number, same with ICG, you fit a model, you make some assumptions, and then somebody gives you a number that says milliliters per minute per 100 gram of tissue. And then I say, okay, give me the technique that you know that works for this, and let me compare it to it. And over the years, we have went from very basic ones to autoradiography, to ASL, to PET, to everything. And in every single one of them, there are a lot of assumptions in it. So if I make the same assumptions, I could give you the same number as well, because I have a line and I can build into my algorithm, it will be transparent. The hard question that I find for me is the same question you pointed out for StO2 as well, because StO2 is a mixture of oxygen saturation in different vessels. So as Gorm was showing, you make an assumption, this is how many in the arterial, how much in the venous, and we build a number, and a long time period was spent to establish it. So we could give a number like that. The hard question that, as a physicist, that bothers me to give you right now is I'm not sure that that number is the right number for what we think we measure. But if we made a population and if we show that it's nicely distributed, it's linear, we could give you a number. I would like to understand it really exactly, precisely, fine, give me the right experiment to do so we can test this. And I think it's the doctors and the standardization people who have to give us these experiments.
No, there's no question. That's why, and that's the key we are discussing here. That's this bridge between physics, technology, physics, physiology, and then what is the use for the doctor? I mean, what could be the benefit for the patient? Because healthcare is about benefiting patients. But I just throw in here that 32 years ago, I published a paper where we compared Doppler ultrasound, because it's pretty easy to measure blood flow velocity in a blood vessel Doppler ultrasound. That was started in the mid-70s, and we did this research in the beginning of the 80s, and we compared to xenon clearance, which is an isotope technique which involves some radiation, that makes some limitations, and I compared Doppler flow measure of flow velocity in vessels in the brain with this isotope clearance, which is a very sort of, you can say, robust technique. And I got one of these graphs. You can imagine here the xenon and here the Doppler, and here is a line and a positive association and a spread. And the conclusion of the paper was that this Doppler, which at that time was used quite a lot because it was a new technique, it was promising, there were many papers coming out. Our conclusion was that really it wasn't the same. But the editors wrote, there was a positive association, so obviously both of these techniques recorded something about blood flow. And also this was not perfect, this association, and she said, obviously because they are different techniques, which does that differently, so how could you imagine that they would be identical? And then she wrote, go ahead and use it for something so we come to understand, I mean, get that method into use and see if you can produce something with it which is helpful. And that's probably what this machine is about. I mean, that is making it into something which actually can get to the bedside and study something either as a research project so we can be smarter in understanding what's going on, or to the bedside where it can help the doctor treating the baby better. These are two different things. So I think we will have to address this now. This question now, the panel first. This machine, what is the sort of step? This is a step one. It's certainly not a step one because it's a step. But let us say this is step one. What is the next step? What is the next step for this machine? Should we go into making it a researcher's tool, or should we go and aim for making this a doctor's tool? Turgut.
So from the perspective of someone who may have to work on it, to make it either one of them, I think to make it an instrument for the doctors, we need to know exactly how and where it should be used. So I have the feeling that there's a gap in there. So we could spend a lot of time, for example, to make a very thin probe, very soft probe, beautiful interface, and then you find out that, no, I should be using this more like a TCD device, which is a handheld, put it and look at it at a given time on the fontanelle or something. Then we should go back. So I would suggest that we should spend time to make it a good research instrument. And this is where we did something which is very interesting. One result you didn't show is that statistically speaking, the numbers we obtained in 2 different sites are equivalent. And this is a big plus for us. It's one of the first times that at least we have built devices which we send out to 2 different centers, and we have data quantitative to say that they are producing equivalent results operated by 2 different groups of people. Question now is, shall we do this 5 of these, or shall we spend 2 more years to make it a bit better? I would hesitate from spending a lot of time to make it a real clinical device yet. I would like to define a good clinical research question to answer the question of how you really like to use this device and where it may actually make a difference.
Even in neonatology, the customers vary quite a lot. Even it is the first 28 days after baby is born, the customer is completely different. And when I see this device, I would say that if the purpose is to improve the outcome of babies below 28 weeks, I can't use that. Because I am amazed, how can I get a good recording of oxygen tissue saturation using this kind of sensor that has to be in a temporal squamma, where you really have here water. So I guess for this kind of population, the brain, for everybody, the brain has a lot of water surrounding, and if is to be very, very well fixed to the skin-to-skin contact needs to be perfect. So you have to work on this firstly, because you need something much, smaller to move to the forehead. Otherwise, you get water measurements, nothing else. If you want to look at term babies, asphyxiated term babies on hypothermia or congenital heart diseases during intervention before and after, I would say that you can use this probe. It's perfect, because here the temporal region would be perfect, stable and so. So if this means that you want to start doing research or use in a clinical setting right now without spending time and money, I would go for the term infant. If you want to go to exploit the technique among the preterm infants, you need to spend time and money changing the instrumentation. This is my first recommendation. And then, for sure, we want to improve the outcome of the infant. So the ideal scenario would be to have a good instrument to use in a routine clinical setting to improve the outcome, continuous monitor. But I don't know if we are still at that point. We need more results to do that. So to present that, this technology as that, under my point of view, currently is too early. So I could go for physiological interventions, which are many, many settings.
So I understand on this side of the table, the idea is to go for a research instrument. Yeah, because this is, I mean, so when we set up this, we said, I mean, I think this was to convince the EU, perhaps. We said this is going to save 15 million babies a year. Yeah, in the long run. Yeah. We never said in 3 years.
Basically, I agree with this. I think you should first go for a research instrument, but then on the other hand, you also have to consider clinical studies. So if you have this information and if you want to improve outcome, you need something to say, okay, we have to keep the flow in a certain range. How can we do that? Does it really help improve the outcome of the infants? I think you need to have a research instrument to address these kind of questions, and then when you can show that the outcome is improved, then I think there will be enough money to make a clinical instrument.
And I think there's always a sort of two competing parts onto which we tend to put the effort on. So most of the criticism that I'm hearing, which I agree with also, has to do with the probe right now. So we have been more concentrating in building a device that provides a very good, highly accurate numbers with the best components out there, with the most stable components out there, and that's a big challenge. So if you look at how this device would have looked in 2011, 2012, when BabyLux started, this whole height would have been filled with electronics and cables running all over the place. So if we say this instrument and the quality of the data you get is good, then we focus separately on the probe. So we have done a lot of improvements on the probe here too, because this is also an industrializable probe. We had a partner who has built a probe that you can replicate many of them. But they are also guys who have never actually put an IRS probe on any subject before. So this is, let's say, a probe version 0.5 with a device version 1 that is running. So this is a very valuable input that we have to work on making a better probe to be used on a premature infant. And we need to sit down with a large group of people to discuss, if I want 5 of these devices to be used in a place, I need to measure it for this many hours, and I can spend 2 minutes to put the probe on. Fine, let's see if we can make the right probe for it. I think this is where we are with this. I mean, this is a competing interests, competing difficulties, and in BabyLux we have advanced a lot in many areas simultaneously.
Absolutely.
So my name is Margarita Brida. I work for industry healthcare in the Siemens Healthineers. So if I just allow a comment or reflection on this point, I have read about the project. We have not been involved, so I have a very, I say, superficial understanding of the details. But at this point, the experience that we have from in industry side is that the point you are seems is the point where you will need an advisory board. So you would need, what we would do in the development phase is when a prototype has been used in only very few selected places, is that actually to open the discussion to more experts or more centers, and by a structured advisory board, which brings then people from different backgrounds, like from different areas of the medical field, and also external people who are not in depth involved in the development of the instrument, but they actually have the knowledge of the clinical environment where the device should be put in. And usually the experience we had, I mean, I have in mind, for example, I'm a physicist from background and worked in R&D for computer tomography. And when I saw the first images from the first CT, commercial CT scanner, I saw them, I mean, I'm not that old that I could be working at that time, but I saw it already using very advanced technology, and I was horrified. I thought this could never be used. This would not bring any benefit. How can you see something in such a horrible low-resolution image? But the reality was that by putting the device in use in the medical arena, in a hospital, in terms of clinical research, you got so many inputs from the use that you could have. And sometimes we are used at the end of an advisory board or by testing your device, used to hear from the physicians, you have no idea what your device can do. I can use it in so many different ways, and I can tell you how much better it can be with very few adjustments. So when the technology at the basis is solid, of course, then there is an explosion, a very fast raise in then going to the next step will be a clinical device, and it can be commercialized. But I understand from what I heard is that would be probably the way to go for the future.
No, I think this is a very important comment, because clearly the physics hasn't changed. I mean, there's no, compared to computer tomography using radio, this is technology and application. So it is application technology, and then the clinical demand, and then more technology, and then more clinical demand, and this is the way I feel it's expanding. And when we speak of flow and oxygenation, these are all over medicine. I mean, not only neonatology, this is, everybody says it's important, but it is not part of our clinical routine. I mean, if people come in with cardiac failure, and what do they have? An EKG and perhaps an echocardiography to show how the pump is pumping, but measured flow, no. Oxygenation, no. And I mean, this is what matters. And the reason why it isn't is because we can't do that reliably. These machines are not there. And this is what this project is about. It just happened. I don't know why this came. Perhaps some of you know why did we strike on neonatology?
So this is a bit about the history of how I started calling people up, I guess, when BabyLux started. So I read the call. As Alessandro stressed many times, it said you shouldn't do research. And it said it should be an area where the technologies you use have already been proven to give accurate results, where accuracy is more defined from the physics perspective. So then I scratched my head and I said, if I try to do something on the adult brain, I'll have to deal with the superficial issues, the systemic issues, and it could be easily argued that it's not exactly well proven. We would have had to put work packages and tasks dealing with that. And we had a lot of experience measuring congenital heart defect babies with Children's Hospital of Philadelphia, and we had a good relationship with Polyme. So I called them up and I said, I want to do this and I want to introduce neonatology. Then I think I told to Martin afterwards, who suggested you. Unfortunately, Switzerland was not allowed in this project, so Martin couldn't join. And this is why we picked neonatology. Whether it should have been the preemies or the term babies and such, we could discuss, but this is my recollection from that time, how I picked that area.
Yeah, so this is a story about things develop because you know people. Yeah, that's human nature, isn't it? But there is also a physical reason, and that is the newborn brain is up here, and it is within a very thin box, whereas a human brain is, and there's something about light having difficulty getting in there. I mean, a little bit of light gets in there, but...
I like how you said the human brain versus the premature brain.
No, the adult brain. And you could say when it comes to light and brain, then the newborn is really a special case. It's a better case, could say. Now, if we were more in the medical side, then I think we could discuss what kind of research instrument do we want or need? Do we need this to do pathophysiology? I mean, this kind of, you study 10 patients in order to find out how a particular blood flow reacts to some drug or something. Is that our problem? Or is our problem really more sort of not pathophysiology, but more practice-oriented? Is it better that we do this or that? Do we need physiology, Agnese? You like physiology. Yeah. Adelina, do we need physiology?
We know a lot about physiology. I would prefer to start doing interventions that we really use in
But you
in
and
a
I are
clinical.
old.
Sorry?
You and I are old.
Yeah.
We did pathophysiologies for 30 years.
That's
and
why.
we think, Monica, you also on our...
Interventions.
I think we need to study pathophysiology to understand what's happening in the brain of these premature infants, because we think we know, but perhaps, but I'm sure we really don't know. But I think that these pathophysiological studies are needed to help us to guide clinical intervention. So as a neonatologist, I feel the need to have an instrument to monitor these premature babies in the clinical part of their life in order to have to have an instrument which can guide me to intervention, because we want to change the outcome of premature babies, and we talk a lot how we could be able to do it. So I think that I will go for studies related to intervention, which can be useful to us. I used to say that I work at the neonatal intensive care, which is lung-oriented. Most of my colleagues are really worried about the lung. We should not spoil the lung and the heart and the cardiovascular stability of the patient. But because it's easier, you have a chest X-ray every day, you do the echocardiography, and when you look at the brain, you just look at the brain ultrasound, which is a kind of picture just telling you that something has happened or hasn't happened yet. But you don't know if something is happening and you don't see it yet. So we need something in between telling us what's happening in real time in the brain of these babies. And if we can do anything to prevent brain damage, because when we see the brain damage with the ultrasound, it's too late. So if we have an instrument which can be used cut side, I think that also all the other colleagues will pay more attention to what is happening in the brain, because you just see that it's desaturating. But the next step is that when I see what is happening, what can I do? So this is the real question. That's why I think we need intervention studies in which we see what's happening and we test what we can do to improve or stop this unfavorable event. And I just would like to make a comment on this project, because I think that one of the main results of this project is the strict collaboration between technical people and clinicians. We entered the lab, Alessandro's lab, and we couldn't understand almost anything at the beginning, and they entered the intensive care unit and the delivery room. And now they realize what we need as a clinician in this particular field of the medicine, which is neonatology. So I remember Alessandro was really impressed in the delivery room when one baby needed to be resuscitated, and he understood what we need in this emergency situation, and they also realized how small our babies are. So I think I did most of the measurements together with Agnese, and I'm pretty aware of the technical problems related to the size of the probe, to the stiffness, but I'm also confident that thanks to this good collaboration and these technical issues can be improved. I don't know if they can be solved, but for sure they can be improved thanks to strict collaborations. So it was not easy to get these results, and it's quite demanding, but I was also positively impressed by the results when we analyzed. So I'm pretty confident also that this instrument can be improved.
Adelina.
Coming back to the question on the studies, I fully agree with Monica. Doctors cannot go forward without physics or engineer, whatever. It's the future of medicine. And I think that in any large hospital, there should be, as a staff, technical people, I mean, no doubt about that. Coming back to the studies, I think that the perfect combination is physiology and interventions. So I am thinking about something that we define for the SafeBus. I mean, physiology, this instrument provides a lot of, could eventually provide a lot of information about physiology and physiological changes according to interventions or the clinical situation of the baby. The critical point to me would be to define a time frame for the observation. That could be one intervention or many interventions. But what is essential is to have an algorithm of reaction. Unless if you don't define a clear pathophysiologically algorithm for intervention, as a reaction of what you really see on the screen, you are not going to strengthen the potential of the technique. So my recommendation would be time frame, whatever, could be 3 hours if the intervention is short, 3 days if you want really to address a question that is relevant for the patient, and develop an algorithm that is pathophysiologically oriented to react to changes in the screen, because if not, I guess you can waste the time. So, and I think this is one of the things that surgeons and anesthesiologists had very clear in relation to the near-infrared spectroscopy technology, is that they really help them because they have clear intervention, clear reactions to changes on the values they get. And that is crucial, to my point of view.
Thank you, Adelina. Then you have 10 minutes left of this roundtable, so I just wanted to address one, or we could discuss one issue, and this is more like a technological way, because this is obviously a product of our level of technology now. If we want to put this into to patients, we need some compliance with some standards and safety and stuff. And in the end, this could go through a procedure being CE marked, which means that it is clear that this is safe and fulfilling. And this is a lengthy procedure, and once you've done it, then you're sort of stuck with that until you sort of, if you change it, you'll have to go through the process again. And since this is a field which technologically developed very fast, we end up, which I think is a fact for much medical equipment, of yesterday's technology. What should we, I mean, again, this is what are the limitations, the challenges and opportunities? Because we can work as we do now with this machine, which is not, I mean, we have an investigator's brochure, Udo, it's something like 40 pages. And in Denmark, we have had people looking at that and examined it medical authorities have been looking at it, and then they've given us permission, and we have had to pay them to do the work in order to allow it to put it to babies. And if researchers have to do that, then they might not buy the machine. If it's CE marked, then, well, we have to sell a number of copies before it sort of pays off.
May I comment something on this? So my name is Udo Weigel, so we are the SME partner in the project. So, of course, right now with the resources that we have, this will be a very lengthy process and a lot of money. But I think what has come out quite clearly is that we would rate it still a research device. So once the application is settled, and once we are happy with the results and the precision and accuracy that we can reach, the last step, of course, with the present resources, it will be a very lengthy process, but then you can get people interested in the technology, and probably you would be surprised how fast it can go. If you can make a sound application case, you can get in the money to bring it to the market. This is product development. I think once the first question is really nicely answered, then of course the speed up of the CE marking depends basically on the money that you put in there. And if you have a really sound case, then the money will come. If you don't have the sound case, then you do it with, let's say, money you can get probably from different sources, and of course that's a delay process that applies that, or that leads to maybe an effect like you said, that it takes a long time between the technology is now present state and you can use it later on as a product, because all this then this approval process with not much money takes a long time. That's a big problem, but I would say if it's very promising in the application, then this is just speeded up by getting invested money into the projects.
Maybe can you give us a rough idea what is much money and what is little money?
So, well, maybe you'll say that in your talk. Yeah, maybe that's part of the things I wanted to address later on in the presentation. But the biggest money goes basically in the clinical trials, and this is something that we are an SME. It's clear that a company like our size, or even something that is 2 times our size or 3 times our size, that's not something that you can provide, or you can basically gain with other products and get to this. So there are basically 2 options. Is you look for public funding specifically addressed to clinical trials, where you provide this money and you would try to get a grant for that. The other point is, of course, if you're very convinced about the application and you can prove it very nicely, and all the engineering shortcomings, all this, I think all the technical part we're always commenting that this is engineering. You can find a solution. The question is, if you put the money in and if you put a little bit of time, the more money, the faster it gets, but you will find a solution. The application has to be really convincing, and then you get the other steps. And then you can also look for private investments to do that step. And I mean, we were trying to get a number out of this. To be honest with the estimates, we got something like 2 to 4 million for the clinical trials, but to be very, very honest, that's more a guess than a calculation. So it's hard to estimate, but the big money goes there.
May I say something? Just a short one. An exercise that I would love to do with clinical friends, let's say, critical friends who are willing to spend time to think about the problem and discuss, is imagine everything you said about that has to be improved is improved. Where would you bet your time and resources to test this machine on? So what you said is correct, so interventions and physiology, but which one exactly? What are the numbers for the impact? And how much of your time and resources would you bet for it? And how many centers would you need for it? So if we know the answer, of course, we can try to make a better calculation on it. And as a physicist, I cannot do that. knowing Udo, who's also is a physicist, he cannot do that, and without doing that, we are always sort of making wild guesses. And I have not had the chance to hear this in the neonatology application yet. Some presentations that I heard from Gorm makes a very good case, for example, for oxygenation, very well determined how many babies' lives might be saved this much later. So I think we have to go beyond that and talk about this.
Yeah. Well, we have a couple of minutes left. This is a chance. I mean, the questions are the most important. I mean, the question is, Nils. Nils is also internal to the project, we have to say.
So, we talked a lot about the probes. So, actually, it was Fraunhofer IPT carrying out the activities around probe design together with the partners and probe manufacturing. So, as Turgut already pointed out, so the system and also the probe were designed from the physical perspective to have the best performance possible. But as you have seen, the best performance is not achieved on babies. And we should discuss also about different levels of requirements for different kinds of probes. For example, we talked about the handheld probe that has been suggested by Gorm. So it's a very promising approach, and I think also makes it easier for us to manufacture it. And the fact that we integrate two different modalities like TRS and DCS also implicates different technical requirements from the fiber side, and also some limitations, I think, in design freedom. So, and maybe it would be an option to have a different number of modalities, maybe decrease the number of modalities for different kinds of probes, maybe having a more flexible probe which does not maybe provide both modalities, maybe only one, and having this handheld probe which maybe implements all possible modalities. So that could also be one strategy to go forward.
Last moment responses or Heidrun?
You were discussing this point about getting stuck when you have to do the CE marking and go to the market. I think it does not tell us we have to stop, or it's end of the life. Things are going on, and when we are looking at the big manufacturers, they are coming up with new products every now and then. The critical point here is I see you have to get to the market at once. And when you are there and when you grow, then you probably can do innovations more and more. But my question, I don't know whether you want to put this question again later, but I think in parallel, you would also do the research in the lab to be on time with the developments and to make up with the current state of the art in science, or go beyond, so for the next version. And that's my question all the time when Alessandro says he wouldn't be allowed to do research. What would you do if you were allowed to do research?
Yeah, 30 seconds.
I can't answer. I mean, it's confidential. Okay. No, I'm joking. No, actually, as said, we are running also together with Turgut and other colleagues, other European project on, I would not say the very same technology, but the physics is the same. So we are developing novel components, devices, approaches, but it is a long run. I mean, it will be at least 5 years from now before we can start to build a device like this, probably. And so in this phase, such a device is really important to get real data from the clinical research scenario that can also be used to foster the development of new advanced technology. So actually, we as a university research center are actually working on both basic and applied research. So we have foot into shoes. So we need to work in parallel in both, because I mean there can be a feedback in both directions, from the clinics to the research and from the research to the clinics. And of course we need money for for both.
Sure, yeah.
Just a 30-second remark. So the one slide that I like showing in my talks is about an instrument that was made here in Milano in the 19th century to measure cerebral blood flow on the head by looking at the tipping of a balance. And what I say at the end of my talk is, we are not just advancing new technologies, we are trying to measure very hard to measure parameters here. And if our patients did not wait 140 something years until people tried to introduce, let's say, imperfect devices to get this data. So we have to move some things into the clinic and work on improving these things. Sure. And these parameters we measure, people are trying to do with many technologies, not just light. And we should always keep this in mind. We are looking at the hard problem using frontier techniques, and the babies have to benefit from it. They cannot wait for us.
Yeah. No, I mean, this will be the end of this, and I'll also add to that, there's no question that this is going to be parallel processes. I mean, so technological development will happen parallel with sort of exploration of medical understanding of illness and in possibilities of intervention, and at the same time the development of clinical practice, so that our improved technology and improved understanding comes to the benefit of patients. And this is clearly parallel processes. Yeah. Thank you very much. This was end of this session.